This recorded session is dedicated to our supplementary discussions on another medically significant group of bacteria, the gram-negative bacteria covering the group of Enterics, Neisseria, Spirochetes, Rickettsia, and Chlamydiae, and Haemophilus, Bordetella, and Legionella. Okay. So the enterics are a group of bacteria that reside animal and human gut. So they're found in the intestines of humans and non-human vertebrates. There are several families or groups within the group, which include the Enterobacteriaceae, the Bibrionaceae, Pseudomonadaceae, and the Bacteroidaceae. So they're part of our normal microflora, of our intestinal normal microflora, and depending on the virulence of the species, they can cause gastrointestinal tract diseases. The Enterobacteriaceae are a family of gram negatives, a very large family of gram negatives, which cover at least 30 or 40 genus or genera with a lot of species within the group. And as the name suggests, so this group are primarily the dominant representatives of the enterics, no? Vibrionaceae, on the other hand, are a group of uh, aquatic inhabiting gram-negative bacteria, both freshwater and saltwater, usually associated with food and waterborne infections. So the type species for this group is Vibrio cholerae, which is known to cause a very problematic disease, no? So, and for the case of Pseudomonadaceae, so this family of bacteria, of gram-negative bacteria, are also a big group, no? They have multiple genera, which include Pseudomonas as the type genus. This group also covers Azomonas, Azotobacter. Okay. So for the case of Pseudomonas, we'll cover a different. We'll dedicate some discussion slides to look at the virulence of some type species under the family. And then we have the Bacteroidaceae, which are primarily associated with um, environmental sources. So they're largely an environmental bacteria. So they're also found in the human gut as part of the enterics group, but primarily, I mean, if we are to compare or to put them side by side with the others, so the Bacteroidaceae are mostly uh, environmental. So they have, in my opinion, they have lesser representatives that can be pathogenic to humans. Okay. Now, in terms of grouping the enterics, there are several factors to be considered. Primarily, the phenotype of the bacterial species, its biochemical profile, its physiological response to environmental conditions, to nutrient availability, to triggers in the ambient, change in temperature, the acidity of the ambient, as well as availability of oxygen. Structurally, we can also classify enterics based on their antigenic phenotype. Okay, so under the biochemical profiles, there are several tests that we perform in order to classify or to do a phenotype profiling of enteric bacteria, such as their ability to ferment specific sugars like lactose, their ability to produce hydrogen sulfide, produce or express enzymes that can break down molecules such as urea and polysaccharides such as gelatin. So for the antigenic classification structurally, so there are three key antigens that we can look at when we're doing serologic classification of the enteric bacterial group. So the O antigen is the primary polysaccharide associated with the outer lipopolysaccharide of gram negatives. So we call it O antigen. O relates with somatic. So O antigen is the somatic antigen of gram negatives within the enteric group associated with the lipopolysaccharide. Then we have the H antigen, which is the flagellin protein, the protein filament forming the filament portion of the flagella. And then we have the K antigen, which is the capsular antigen. Okay. Now, in terms of their clinical significance, enterics primarily cause two major infections or diseases. So there is diarrhea, which is which may or may not have systemic invasion associated with it, and other infections, non-diarrheogenic infections such as urinary tract infection, pneumonia, presence of bacteria in the blood, especially in cases of patients in hospitalized or healthcare settings. No? And there are several manifestations of the infection and disease caused by enterics. So first, diarrhea due to enterics may not have cell invasion, and primarily the route for causing the disease sequelae is due to the release of toxins. No? So we have exotoxins, which are secretory bacterial products. They're released by the bacteria during the invasion phase, the colonization phase. So exotoxins can translocate inside the host cell, can covalently alter the structural integrity of the cellular components, and may cause cell death, no? Or can lead to cell death. So they're cytolytic. And we also have enterotoxins, primarily targeting intestinal cells of the gastrointestinal tract. I think I've explained an example of this during our on-site lecture on enterotoxigenic E. coli, the heat labile the heat stable STB toxin of enterotoxigenic E. coli, primarily altering fluid electrolyte balance of intestinal cells. So examples of those enterics that can cause diarrhea without invasion, without cellular invasion, include the ETEC, the enterotoxigenic E. coli, and Vibrio cholerae. Okay. Then there is diarrhea that is associated with invasion of the host cells, so the intestinal epithelial cells. Cell death can be an approach, or can be a route where it will yield to, or will result to, RBC leakage into the stool or the host. And cell penetration, primarily associated with toxin release, associated with inflammatory response. The WBCs infiltrate the location of infection and can cause symptoms associated with it, such as fever. An example, or some examples of enteric agents causing this kind of diarrhea with associated invasion of the host cell includes EIEC, E. coli, Shigella, and Salmonella. Okay. Then there's another kind of diarrhea associated to enterics, more of a deeper invasion, because this time it goes beyond the GIT, attacking lymph nodes, and we become more distributed via the bloodstream. So its deeper invasion can result to systemic shock associated with symptoms such as fever, headache, pain in the abdomen area, elevated counts of white blood cells, and diarrhea associated with leakage of red blood cells and white blood cells. Okay. Additionally, this kind of diarrhea can result to mesenteric lymph node enlargement, can also cause bacteremia, the presence of bacteria in blood, and sepsis, no, which is the associated, or which is the response of the host, the immune response of the host due to the invasion of the bacterial pathogen. Some examples of agents causing this kind of enteric infection, enteric disease, are Salmonella typhi and Campylobacter jejuni. Okay? Now let's look at the different families of enterics, and let's have a review of their characteristics and virulence determinants. So first we have the Enterobacteriaceae family. The type species or most common to cause infection is Escherichia coli, and its different toxigenic, non-toxigenic groups, no? Commonly causing infections in women of the infections of the urinary tract in women, can also cause meningitis in newborns, can cause gram-negative sepsis, and also hospital-acquired pneumonia is associated with E. coli infection. Now, there are several pathogenic serotypes of E. coli. So we have under enteric E. coli, so we have enteropathogenic E. coli, EPEC, which can cause diarrhea in children, commonly in children. Then there is enterohemorrhagic E. coli, which can cause hemorrhagic colitis or hemolytic uremic syndrome, no? Leading to kidney failure, may lead to kidney failure. Then that's EHEC. Then we have enterotoxigenic E. coli associated with traveler's diarrhea. No? Then enteroaggregative can result to diarrhea in children. Associated or related symptoms include diarrhea with mucous secretions or mucous material rather, and vomiting. Unlike in EPEC, where the diarrhea is more watery. Then we have diffusely adherent E. coli, which can cause acute diarrhea in children. The symptoms include watery diarrhea and recurring urinary tract infection. And we have EIEC, enteroinvasive E. coli, which is shigellosis-like and the kind of disease that is produced. Also associated with watery diarrhea and dysentery. So we're looking at the virulence factors. So some of the virulence factors I would like to emphasize. So we have Shiga toxins or Shiga-like toxins for EHEC, for enterohemorrhagic E. coli, which are expressed by genes associated with Shiga toxin production. So for Shiga toxins, so these toxins can contribute to the sequelae of hemolytic uremic syndrome and thrombocytopenic purpura. Okay. Aside from EHEC, no, Shigella, no, can also be produced by Shigella can also produce Shiga toxins. Then we have for the case of diffusely adherent E. coli, the AIDA proteins. So AIDA proteins is an adhesive or adherence protein rather. So allows for the attachment of the of the bacterial cell to the host cell. Then there is AAF. So AAF is associated with the fimbriae. It's a protein, um, attachment protein of the uh, EAEC group of E. coli. So primarily with their fimbriae. Okay, so AAF stands for aggregative adherence fimbriae. Yeah, so it's a protein for adherence of the pathogenic serotype identified as enteroaggregative. Okay, now let's look at other species. We have Salmonella, which is a foodborne, causing diseases such as typhoid fever, gastroenteritis, and also sepsis. Then we have family Vibrionaceae, primarily aquatic inhabiting. So both freshwater and saltwater. No? So diseases include cholera, gastroenteritis, duodenal ulcers, and chronic gastritis. Then we have Pseudomonadaceae, the type specimen, aeruginosa, forms fluorescent pigments, highly known to be resistant to a lot of antibiotics. It's a notorious biofilm producer. Its capacity to produce or secrete a biofilm or aggregate as biofilms actually give it an advantage against multiple types of antibiotics. So, some of the diseases caused by pseudomonads include pneumonia, sepsis, UTI, and infections in areas that are non-intact, such as burned um areas of the skin. Then we have the Bacteroidaceae. Okay, example is Bacteroides fragilis, which is an obligatory anaerobic. It can, when distributed in other areas, can cause abscesses. Then we have the Neisseria. So Neisseria are gram-negative cocci. They have pili and they're aerobic. So looking at the appearance of their cells, their kidney bean-shaped diplococci. They grow best in environments that are rich in carbon dioxide. So there are a number of species, but those that are pathogenic to humans are Neisseria meningitidis and Neisseria gonorrhoeae. So gonorrhea is an STI, can cause an STI, while meningitidis is primarily affecting primarily affecting our nervous system, central nervous system. So let's look at the characteristics of of some of these species. So let's first look at meningitis. So Neisseria meningitidis, also known as meningococcus. Uh, by the way, I forgot to mention, no? So Neisseria are colonizers of the mucosa. So they're primarily colonizers of the mucosa. So you can expect in terms of its epidemiology, its distribution, that they are normal parts of the they're parts of the normal microflora of the mucosa. About 5% of the population are carriers of N meningitis, and their transmission is primarily via the respiratory route, secretions of the nasopharynx. So some of the virulence factors of meningitis, Neisseria meningitidis, include secreted capsule polysaccharide with different serotypes A, B, C, can cause meningitis and inflammation of the meninges. Also, an endotoxin is associated to N meningitis. So the endotoxin of its lipopolysaccharide can be released in the bloodstream, can cause blood vessel destruction, no? And lead to hemorrhage and eventual sepsis. So additional virulence factors of the species include a proteolytic enzyme targeting cleavage of immunoglobulin A, so IgA protease. Additionally, it can sequester iron from the human transferrin protein In terms of adherence and colonization, it's pileated. So for adherence, so the pili serves that function. Now for the cause or the infections that are caused by meningitis, we have bacteria in the blood. We also have meningitis, infection of the inflammation of the meninges. And fulminant meningococcemia which is usually a fatal kind of sepsis. It's associated with purpura, with eruptions of blood vessels. And lesions are generally purpuric, can lead to necrotic in a particular state, no? And in the nasopharynx, no, as initially they inhabit nasopharynx, no, can invade the bloodstream, no. So in fulminant meningococcemia, hemorrhage, hemorrhagic eruptions, low blood pressure, hypotension, and cardiac depression can happen. There's also rapid enlargement of petechiae and some purpuric lesions. It can happen so fast, no? Those symptoms or those sequelae can happen within hours of initial presentation of the infection. So here you can see meningococcal infection, the petechial rash, which are pin-like in appearance and eventually can become necrotic and hemorrhagic. How to treat meningitis? We have several antibiotics such as penicillin G, ceftriaxone, and rifampin, which can be given to patients that are close contacts of the initial patient. No? Ceftriaxone target cell wall synthesis. Rifampin blocks RNA synthesis. Then we have the second species of Neisseria, which is Neisseria gonorrhoeae, no? Also known as gonococcus. So it's an etiologic agent of an STI, the second most common STI, STD. So some of the virulence factors also are similar with meningitis. So they are also pileated for adherence, proteolytic enzyme IgA targeting IgA protease, which is proteolytic, targeting IgA. Then there are outer membrane proteins of Neisseria gonorrhoeae, protein 1, which is a porin for the production of or allows the structural attack of the host cell membrane. Okay, and then we have protein 2, which is an opacity protein, no, primarily for adherence. Okay, so protein 1, PI, for Neisseria gonorrhoeae is actually variable. There are different classes or groups of it that are already known. So protein 1 in Neisseria gonorrhoeae, I've mentioned the primary function of it as implicated in the pathogenesis of the species. It can translocate from the outer membrane of the bacteria into the target cell membrane of the host, affecting functions such as phagocytosis, okay, for the production of phagosome. Additionally, protein 1 can also cause hybrid porins. So these hybrid porins, they can assist in the they can assist in the structural destruction or integrity issues of the host cell membrane of the target cell membrane of the host cell. Then in terms of the infections they cause, and gonorrhea can cause diseases in men and women. So in men can cause urethritis, then also complications such as inflammation of organs such as the epididymis. Here you can associate with the secretion, or you can see the associated secretory material produced if a male host is infected with gonorrhea. In women, can cause painful burning during urination and also purulent discharge, similar with that of men. It can cause or it can lead to PID, pelvic inflammatory disease, which is characterized by the infection of the upper reproductive tract of women. So organs such as the uterus, ovaries, fallopian tubes are affected by PID. And if untreated, or if treated late in the game, no, it can cause complications such as sterility, the inability to produce or to bear a child, ectopic pregnancy, and abscesses in the muscular area of those reproductive structures. Okay? In neonates, in newborns, and gonorrhea can cause gonococcal disease, that can result to blindness of the newborn, ophthalma neonatorum. The blindness or blindness as an end effect is associated with damaging invasion or the damaging invasion of the infection on the cornea of the newborn. Okay? For treatment, so we have several antibiotics such as ceftriaxone and doxycycline and azithromycin, which are targeting protein synthesis. Okay? I'll divide this lecture into two. So this is part 3.1.